Hello students, I hope you are all doing well. Welcome to the class. I am going to start 10 questions again for molecular basis of inheritance as I have told you in my previous video also. Right, every day we'll be doing a practice of 10 MCQs here for our upcoming NEET 2025 and some questions will be helpful for your board MCQs also. Right, so be very focused and try to learn. See, first question, they are asking that there's a short piece of DNA which has 20 base pairs, right? In board exams, in case study questions, they can give any such kind of questions, right? So see, 20 base pairs are there in a short piece of DNA, right? And we have analyzed the number of nucleotide bases, sum of the number of nucleotide bases in both the strands, right? And we have obtained the result. Strand 1 contains 4 adenine and 4 cytosine. Guanine, thymine, we have not, you can say, analyzed how many are there. Strand number 2, we have analyzed that 5 cytosine are there, but the rest 3 we have not analyzed, right? This is the meaning of question. Then the question is asking that in strand number 2, how many adenine will be present? So I hope you know the meaning of complementary base pairing, right? If you have learned this chapter, you know very well. If you don't know, then in biomolecules also you have learned, right? So we cannot say that we don't know this, but then also a quick recap: adenine always base pairs with thymine or vice versa, right? By 2 hydrogen bonds, and guanine always base pairs with cytosine by 3 hydrogen bonds. So it means that if you know the sequence of one strand, the other strand of the DNA can easily be guessed by checking the complementary bases, right? If you know that, okay, I have 4 adenine in strand number 1, then just tell me the total how many thymine should be there in strand number 2? 4, right? Because 4 adenine must base pair with 4 thymine in strand number 2. Okay, if you have 4 cytosine in strand number 1, how many guanine residues should be there in strand number 2? 4, right? Correct? Now, if you have 5 cytosine strand number 2, how many guanine should be there in strand number 1? 5, right? So, out of total 20 bases, okay, sorry, 20 base pairs, total how many base pairs are there? 20, right? In which how many you obtain the results? 4 plus 4 is 8, right? Plus 5, 13. So 13 base pairs you calculated, right? How many are left? 7, right? And how many base pairs are left here yet to write? 7 only. So it means that 7 must be thymine, and as a result complementary 7 must be adenine in strand number 2, right? So you understood how to find the answer. Answer should be option number D, right? 7 adenine residues must be there in strand number 2. You understood how we have done this, right? Total base pairs we already know as the information is provided in the question itself, right? Rest all we guessed by the complementary base pairing, right? And maybe you remember that this is the hallmark feature of DNA double helix model given by uh, Watson and Crick, right? So on same concept you can solve this question. Okay, I hope it is clear. Now question number 2, right, which is also very good question. Right here they are asking that what is the smallest part of DNA molecule that can be changed by point mutation? Remember about point mutation? You have learned as an example point mutation. Which example you have learned? Everyone must write in the comments. It is sickle cell anemia, right? Sickle cell anemia is example of point mutation. Okay. Now in sickle cell anemia, you remember that if you have this particular triplet or codon, okay, GAG, it was replaced by GUG, and the change happened at the 8th position of the you can say your nucleotide sequence, right? So 8th position base was changed. Instead of adenine, uracil was there, and this is mRNA sequence which I have written, right? Complementary to this you can understand the DNA, right? But codon word generally is used for mRNA only. It is not used for DNA, but because these triplets are coming as a result of DNA transcription into mRNA, we can consider it there also, right? So this is the meaning of point mutation. Okay, here what happened? Substitution. Okay, substitution of A with U. Okay, So if any base pair, one single base pair is substituted with some other base pair, then it is called point mutation. At one single point, change happened, right? They are asking that point mutation can happen in which of the part of DNA which is smallest, right? One by one, I will tell you. Okay. First, if you consider, they have given option as oligonucleotide. What is the meaning of oligonucleotide? Anyone? Can you tell me in comments also? Oligo means few. Okay, few. And nucleotide, you know very well. So few nucleotides, they make oligonucleotide. Maybe you have learned a familiar term which is called oligosaccharide. So in biochemistry, usually we use this term, right? It is not directly given in CRT. Oligonucleotide means few. Okay, very few base pairs. Let's suppose 50 base pairs. Actually, it is not this much also. It should be very less. So let's suppose 10 base pairs, or sometimes when you will see it can reach up to 50 base pairs also, right? So very few nucleotides when they are present together, it is called oligonucleotide, right? Then okay, codon. Codon contains how many nucleotides? Please tell me. If I want to say one codon contains 3 nucleotides or not? It's a triplet, right? So is it smallest unit of your DNA? No, right? So these two options, so already you have to eliminate, right? Then let's talk about gene and nucleotide. Before that, I will tell you one hierarchy. Okay, you can say more maximum organized unit of your genetic material is chromosome. Correct or not? It is maximum organized. Okay, packaging of your genetic material is highest at chromosome level, right? So this is maximum organized. Okay, if I want to write little less than chromosome, okay, I can write chromatin. Correct or not? I can write chromatin here. Okay, which is that particular. What is chromatin? You can write in comments if you know. Okay, chromatin. And chromatin actually is made up of DNA, double-stranded DNA. Right, if I attach histone proteins, I can make chromatin. Then if more proteins like non-histone chromosomal protein, then I can make chromosome, right? So like this is the level of your genetic material packaging. And your DNA is a polynucleotide. Okay, now your double-stranded DNA in turn is made up of what? You have learned what is the definition of DNA. It is a polynucleotide, right? So it means it is made up of so many nucleotides. So what is the level here? Smallest level is nucleotide. Now you must be thinking, where is gene, right? Because in option number C you can see gene, right? So you tell me the definition of gene, please. What is the definition of gene, if you remember? Gene means, there are so many definitions of gene, right? So gene, if I consider here, gene will be made up of, you can say gene is made up of fixed number of nucleotide. Fixed number of nucleotide, right? Like how you will define gene? You will say that this part of my DNA is a gene, right? Which starts from this nucleotide, ends at this nucleotide. Means gene is made up of few nucleotide, which is larger unit, right? Like you can consider that, okay, gene is present on DNA and then you can write nucleotide. Like here you can do like this. You have to analyze it like this. But I am not writing it there because not necessary, right? So I explained it separately. So ultimately, what is the smallest unit of, you can say, genetic material? Smallest, where you can change something and because of that some point mutation can happen, right? So that is this smallest. Okay, what they have mentioned here, smallest part of DNA, right? This will be your smallest part of DNA, okay, where you can do substitution mutation, or you can say point mutation. Here you are going to do point mutation. Then something will reflect at chromosome level also, right? So for second question, what is the answer? Answer will be option number 4, nucleotide, right? Why not gene? You understood, because gene is a part of, like it's a made up of some few nucleotide, right? That's why. Okay. Now, third question. In bacteria, the transfer of genetic material from one bacterium to another through direct cell-to-cell contact is called. Maybe you will be saying that this question is not in NCRT, this is out of NCRT, right? But you should know all these things, right? This is some parts are there in class 11th, which you have learned in biological classification chapter, and some parts are there in this chapter itself, where you have learned Griffith transformation principal experiment, right? So what was transformation? Transformation was, remember, uh, last class also I have got one question which was having S strain, R strain, right? So what was happening there? S strain was capable to do a disease, like it was a pathogenic. It was responsible for a disease called pneumonia, right? And R strain was not having any particular virulence power, because it was not having any, you can say, mucus layer which was responsible for that disease, right? So what was happening when R strain and S strain, okay, they were together, right? And you have taken which strain as a heat killed strain, please write in the comments. You have learned that Griffith has conducted an experiment, and in that experiment, in last of those experiments, what he has done, he has taken heat killed S strain, right? And he has taken alive R strain, right? And when after being killed by heat, what happens to the bacterial cell? Bacterial cell will get disintegrated, right? Only some parts of DNA will be there, that also will become single stranded, but whenever the temperature will become normal at room temperature, right? Again, because the DNA is such a stable molecule, it will again try to do hydrogen bonding, right? And then that DNA will enter inside R strain bacteria, and it will transform that R strain bacteria into S strain. So what happened here? Bacteria has taken up the DNA from the surrounding medium. This is called transformation, right? Is the cell directly contacting? Is there any cell to cell contact here? No, right? So this cannot be the answer, right? No cell to cell contact in transformation, right? What about transduction? Transduction is the transfer of DNA in between two bacterial cell with the help of one intermediate or vector. Like work is like vector, name is not vector, okay? Like a mediator molecule, okay? Two bacterial cells are there, okay? A bacteriophage will, you can say, enter, like it will make its DNA enter inside the bacterial cell while making new viral particles. By mistake, some of the part of the bacterial genome also enters in the viral particle. When that will get disintegrated because of that viral multiplication inside it, again they will be going to the bacterial cell, and they will be infecting the bacterial cells, right? So what will happen? If such a viral particle which has already taken up the bacterial genome, right, and it will again, if it is going to infect another bacterial cell, what is going to happen? It is going to it has taken the genome of one bacterial cell, and it is going to give it to another bacterial cell. Understood? This is called transduction, right? जैसे कि you all know that if you are ordering something, right, you want to eat some your favorite food, so what you will do? You will make order, right? One person will be there who will be picking up the food, and it will be providing to you, right? This kind of work has been done here also. One bacteriophage virus is there. It is going to take the DNA from one bacterial cell and is going to deliver it to another bacterial cell. Any cell to cell contact of bacteria is there here? No, right? That's why this also cannot be considered as correct option. But if you consider conjugation, conjugation is what you have learned in the previous class, right? In class 11th, you have learned about this concept which is called conjugation. So in this, let's suppose this is one bacterial cell, right? And this is, let's suppose, another bacterial cell, right? And what I have drawn there, that is pili, right? Let's suppose this is the plasma membrane. Otherwise, you will get confused that it is extension of cell wall, no, or extension of plasma membrane, no. Okay. So this is our two bacterial cell. This bacterial cell is having its own DNA, this is having its own DNA, but this bacterial cell is having one more thing which is called plasmid, okay, which is smaller, right? This doesn't have this. So this bacterial cell is called F positive. This is having a plasmid called fertility plasmid, right? So this can act like a donor, right? This does not have that plasmid, it will be considered as recipient, okay? And what is this? Who will tell me what is the name of this structure? These are called pili, right? Okay, what will happen here? These two bacterial cells, okay, the pili are hollow tube-like extensions of bacterial cell. they will fuse together, they will make a tube which is called conjugation tube. And with the help of this conjugation tube, what will happen? Okay, the DNA which is present here, okay, it will be copied and it will be delivered to this bacterial cell after getting copied, not the same DNA, right? So here, can you see there is a cell to cell contact? Cell to cell contact is there because of what? Because of conjugation tube. Okay, because of conjugation tube, there is cell to cell contact, right? And this method of genetic material transfer from one bacterial cell to another bacterial cell by direct cell to cell contact is called conjugation. So this is our answer, right? By replication, they will just replicate their own DNA. They will never transfer it to some other bacterial cell. Understood? I hope it is clear. Okay, now what does the repressor protein bind to in the lacoperon? Okay, so they're asking that what is that place where the repressor protein has to bind in a lacoperon? So if you remember lacoperon concept, very easy. Yeah. Okay. This is, let's suppose, a part of our DNA, right? Not our, sorry, it is in E. coli, right? This is promoter. Okay. This is a gene I, which you all know. This is for repressor gene, right? Then you have one more promoter, you have one operator, and then you have structural genes, okay, which are gene Z, Y, and E, right? This is the transcription, you can say, this is the operon which is given in your NCRT, right? So what is the work of this gene I? This gene I is going to make messenger RNA, okay? This gene I is going to make messenger RNA for repressor protein. And after that, this will make a repressor protein. Right? Now, just you have to tell in this question where this repressor protein is going to bind. Let's suppose I am drawing it with this green color. So you know very well this repressor protein will go and it will cover the operator. It will come and bind here. Correct or not? So that the transcription of Z, Y, and A should be very less, right? Because the promoter for these three genes are now having obstruction of repressor, right? Operator should be clear if Z, Y, and A synthesis should be very high. Rate should be very high, right? So you got the answer. Where the repressor protein is going to bind in the lacoperon? Operator. Okay. Now, the central dogma of molecular biology states that this is very easy question. Who proposed central dogma of molecular biology? Please tell me in comments. It is Francis Crick, right? So, central dogma of molecular biology suggests what? Very easy, right? You remember? You have learned that the genetic information in all the organisms, it flows in unidirectional method, right? Except in viruses. Viruses does not come under living organisms. So, except that case, if you consider all the living organisms, right, genetic information flows in a single direction. Okay, from a DNA, we can make one more DNA by which process? Replication, right? From DNA, we can make RNA by which process? Transcription, right? From RNA, what we can make? Protein, right? What is the mechanism? Translation. Okay, this if you know, then you can find the answer very well. This is called central dogma of life, which connects every living organism on the planet. Every living organism follows this only. Okay, the flow of genetic information is which type here? Unidirectional. Can it ever change in living organisms? No, never. Okay, but is there any exception to this? Yes, it exists, but in viruses which are not living. DNA is transcribed into RNA, which is then translated into protein. Correct, right? Now, one question I will ask, please write in comments if you know the answer. It will be very, very helpful to assess that what you are learning or what you are reading. In the next video, I will give you the answer. Question is, why viruses are not considered as living? Correctly, please tell me. Okay. Which of the following is not a type of RNA involved in protein synthesis? Good question, right? Options just check. Messenger RNA, transfer RNA, ribosomal RNA, and regulatory RNA. Now, have you learned anything like regulatory RNA till now? No, right? But there are certain regulatory RNAs which are present but which are not in our syllabus. Okay, so simply what you can do, you can eliminate this option, right? You have learned about messenger RNA or not? Tell me, without messenger RNA, can you make protein? No, right? Messenger RNA is very, very important because it acts like a template for protein synthesis, right? It acts like a template for protein synthesis. Code exists in this, right? Code for translation is present in mRNA. So you require it. Okay? Check. tRNA do you require? Yes, obviously. It is an adapter molecule, right? What's the meaning of adapter molecule? Meaning of adapter molecule is this is going to bring amino acid. Remember the structure of tRNA, the simplest structure if you consider. Okay, if I draw like this, okay, here at three particular positions you have a specific type of nucleotides which are called which make anticodon, right? And at this 3' OH you will be having amino acid acceptor arm. So your amino acid will be attached on this arm and your anticodon arm will be reading the code on your messenger RNA by complementary base pairing, right? So definitely you require tRNA. What about ribosomal RNA? Do we need ribosomal RNA? Definitely we need, right? For what purpose we need ribosomal RNA? Ribosomal RNA makes ribosome, right? And ribosome is something which acts like protein factory. Without ribosome, can we make protein? No. And one of the ribosomal are very, very necessary for us. Why? Because it acts like ribozyme, right? It is that if it is prokaryote, it is 23S ribosomal RNA, right? If it is eukaryote, it is 28S ribosomal RNA, right? Which helps in peptide bond formation when your amino acids needs to be connected. Okay, so that's why it is called peptidyl peptidyl transferase. Okay, it helps in building peptide bonds between the amino acids during the translation. Only thing which is not needed is this regulatory RNA, which are not in our syllabus. So you just simply have to eliminate this. Right. Now, the genetic material in viruses can be. This you know very well, right? Viruses, they can have DNA also as their genetic material, as well as they can have RNA also, but never together, right? DNA containing viruses, anyone? Bacteriophage, you remember, right? Which one? Lambda phage. Lambda phage is having this double stranded DNA, right? Here, QB bacteriophage, remember? QB bacteriophage. Okay, then which else? Tobacco mosaic virus you have learned, right? So yes, in viruses, either DNA or RNA both can act like genetic material. Okay, now see this question. In this question, what they are saying, DNA replication is said to be semi-conservative because. Okay, what's the meaning of semi-conservative? Actually, this was given by scientist Messelson and Stahl. Two scientists are there who actually proved that DNA replication is semi-conservative. So let's suppose you are having this DNA, right? And you consider both the strands are parental strand. I am denoting them as PP, right? So if I want to make DNA, like new DNA out of this, both will act like template. At a time, both will act like template and both the strands will unwind. They will open. Okay, both will act like template strands. Okay, both the strands will be template strand. Utilizing them as template, new strand will be formed here. Okay, using them as template, new strands will be formed here, right? Up, see here. After this, okay, when this will continue to full length, what will happen? You will get a DNA, okay, in which one strand is parental. One strand you just made now, right? So this yellow one I can write this is parental, and the green one is new or daughter, right? Can you see this is considered as semi-conservative mode of DNA replication? What is the meaning? Half half of your DNA will be parental always, and half of your DNA means one strand could have written one strand of your DNA is parental, one other strand of your DNA will be new or daughter, right? So by this what you are able to understand, half conserved, right? Always, whenever this will happen, one strand will always be, you can say parental, and one strand will be new. So this is semi-conservative mode of DNA replication. So what is the meaning here? One strand of DNA is newly synthesized while the other strand is conserved. That's why it is called semi-conservative. Both the strands are newly synthesized? No. Both strands are conserved? No. One strand is degraded? No. Can I say both the strands of DNA act like template strand during DNA replication? And can I say the same for RNA synthesis also, that both DNA strands act like template for RNA synthesis? Please write in the comment section. Okay. Then genes are monocystonic in eukaryotes, but these genes are called split genes because of what? Right? Only exons appear in the processed mRNA. Exons of same gene are located at different locus or loci. Exons are intervened by introns, and introns do not appear in matured mRNA. Okay. First of all, if you consider genes, okay, those genes which are having coding functions, okay, or better I can write those genes which code for protein. Okay, there are many genes in HGP you have learned which don't have any direct coding functions, but there are genes which codes for protein. So such kind of genes which code for protein, they are called cistrons. They make actually cistron, right? Cistron. अब cistrons are of two types. If you remember your text, right? One is called monocistron. So genes can be monocistronic. Okay, I'm writing monocistronic. Okay, and then there are genes which are polycistronic. What is the meaning of these two terms? Polycistronic and monocistronic, these two terms will tell us that from a single transcription unit, how many proteins you can make, right? If I talk about monocistronic, so you have to remember one transcription unit gives one protein only. Okay, from one transcription unit, if I am only able to make one protein, it is called monocistronic, and this happens in eukaryote. This is there in our case, right? But from one single transcription unit, if we are able to make more than one protein, okay, if we are able to make more than one protein, that is called polycistronic, and this happens in the prokaryotes. Okay. Up this polycystonic condition is good. It does not have any problem, but monocystonic condition has some complications. Okay. What are the complications here? Complications which are present here are presence of non-coding, presence of non-coding sequences in between. Okay, I cannot write on that side, right? My video will be coming there. So, no issues. Okay. Okay, now fine, right? Oh, yo. Okay, now good. Okay, so presence of non-coding sequences between coding sequences. Such kind of complication is not at all there in prokaryote. They have only coding, right? What's the meaning of these non-coding sequences? Non-coding sequences are also called introns. the meaning of coding sequences? Coding sequences are also called exons, right? So let's suppose if I want to draw one DNA, okay, uh, or you can say mRNA I will draw here. You already have learned all this, right? In some of the class, if you don't know, tell me, we will discuss that. Let's suppose I draw one transcription unit like this. For clearance, I will change the color, yes. Okay, let's suppose I created one messenger RNA from DNA. Okay, this much area is having information to make protein. This is called exon. And this is first one as per sequence, so I am writing exon 1. This also has information to make protein. This also has. So we have like exon 1, 2 and 3. But in between, there is some part of my RNA which does not have any information to make any protein. This is called intron. This is intron 1. This is intron 2. So what to do? You have to cut these introns or not. You have to cut these introns, right? You need to cut these introns. Okay. So this messenger RNA requires modification. Modification required. And the type of modification which you are going to do here is called splicing. What is splicing? You will cut all the introns, right? And you will join back all the exons together, right? Then your messenger RNA will be ready, right? Then your messenger RNA will be ready to move out from the nucleus. And better I should write, this is heteronuclear mRNA and this is mature messenger RNA. Or in NCERT, they have followed only heteronuclear mRNA and you can say normal mRNA, right? This does not have any introns. This is not having any intron here, right? So this kind of arrangement occurs in the DNA, our DNA, right? That's why it is called split gene arrangement. What is the name? This is called split gene arrangement. Why split gene arrangement? Because the information is not continuous. We have split information, right? We have broken information to make protein, right? So genes are monocystronic in eukaryotes, but these genes are called split genes. What is the most probable option which you can select? Exons are intertwined by introns, right? This kind of sequence is there in our DNA, but whereas I have written here mRNA sequence. So you have to understand same complementary base pairing ke through you can understand it. Okay, so this is the concept. Now last question for the day. Overlapping of codons present in genome of. Okay, this question is little bit out of NCERT, but I want to add some odd questions also so that you can understand overall concept of the chapter. Okay, you have learned one thing, that code is non-overlapping. Okay, or let's suppose if you have learned about salient features of genetic code, right? So in that, remember, you have learned so many features. So out of that, if you consider this overlapping, what is the meaning here? Let's suppose I'm writing here AUG, then I'm writing triple U, then I'm writing triple A, right? Then I'm writing triple G. Like this, for simplicity, I have mentioned like this. It can be anything, right? So when your ribosome will come and read the code, it will read this as individual consecutive successive 3 triplets. Okay, this only should be the reading frame of your ribosome. It should never do like, okay, AUG I have read, now UGU I should read. No, right? Or GUU I should read. No, overlapping is not there. No overlapping is there for ribosome to read the code in your mRNA. It always reads code in triplet format. Okay, one triplet, then next triplet, then next triplet, right? No overlapping should be there. But this is not followed by virus. Virus is exception to this. Okay, so plant, animal, humans, any other organism, living organism, they follow this. They, in our case, the ribosome will be reading the code in non-overlapping manner, right? Three, then next three, then next three, then next three. But overlapping is possible in viruses, right? That's why viruses are very dangerous, right? So this is all for today. I hope you have understood many new things from this discussion. And please support, please come back, please write what you want, please you can say refer your textbook, prepare very well, be motivated, right? And we will meet in next session. Thank you.